All right, for the final part of this chapter, we are going to cover the regulation of the tryptophan operon. So we're switching to a completely different part of the chromosome, and this is a part of the chromosome that contains a number of genes that are necessary for creating, for the biosynthesis of tryptophan. So we know that E. coli can produce, and most bacteria can produce enzymes that allow the biosynthesis of all of the amino acids and essential macromolecules. So they can take a sugar, and eventually that sugar is converted to threonine, tryptophan, histidine, alanine, etc. And so these genes, they're specific genes that are responsible for the production of all of these amino acids. And so again, the general philosophy that you can assume how a bacteria works is that bacteria only transcribe things when they're needed. And so in the case of something like tryptophan biosynthesis is that, there's all these enzymes that are necessary to produce tryptophan, but you don't want to waste energy making those if the bacteria doesn't need them. So why might a bacteria not need tryptophan? So one possibility is already created a ton of it. Another possibility is that it finds itself in a region where it's just able to import tryptophan directly into the cell. So when that bacteria grow in rich media, there's tryptophan already available from it that it can get, simply by by importing like proteins that are in the environment and then cleaving those proteins into the constitutive amino acids. And so if the cell doesn't need tryptophan, it's not going to create the enzymes that are necessary for it. And so again, the sets of enzymes that are necessary for producing tryptophan, they all are next to each other on a chromosome, and they're all regulated together as part of an operon. So this is called the TrpOperon, and in this case there's 5 different structural genes that are necessary for the biosynthesis of tryptophan. And then again, we also have regions of the promoter region that we talk about, and so one of these is the promoter region. There again is an operator site where a repressor can bind to, and then for this particular one, we also talk about a specific part of the mRNA. This is part of the DNA. So the +1 site, the transcriptional start site, is right here. But this results in the production of a leader sequence and a attenuator sequence that does not encode for the protein. So we talked about before, mRNAs have an AUG where a gene starts from, but there's also this region of the mRNA that we've called before the 5' UTR. So this is part of the mRNA that does not encode for protein. But in this case, in the case of the tryptophan operon, the TrpOperon, it's actually very important for its regulation. And we're going to see how that is so in a little bit. But first, we see some very classic negative regulation of this operon, and that's because there is a repressor protein. So this is again a completely separate gene, and there's this repressor gene that's constitutively produced, and this can bind to the operator region to prevent the expression. And so this, and then the way you tie tryptophan biosynthesis into this is that you actually have a binding site for tryptophan on the TrpR repressor protein. So if tryptophan is around, if you've produced a bunch of tryptophan, if you're importing it from the environment, then this repressor protein is bound to the cofactor, and this cofactor is able to bind to the operator to prevent its expression. So this is almost exactly the same as the lac repressor, again with the distinction that the DNA-bound form is bound to the cofactor. And so in order for the repressor to bind to the operator site, the cofactor must be around, the tryptophan must be around. And so when do you need these enzymes? You need these enzymes when tryptophan is not around. So if tryptophan is not around, it's unable to bind, and then you produce all of these different factors. However, it was discovered tryptophan also, the TrpOperon can also be regulated by a process called attenuation. And so attenuation is not actually a modification of the initiation of transcription, but it's actually a modification of the extension of transcription. And this has actually served as kind of a generic principle and the discovery of something called riboswitches. So the reason this is possible is that this TrpR structural genes are preceded by a leader sequence that contain a regulator site called an attenuator. And the transcription of this leader region occurs even when the operon is repressed in the presence of tryptophan, and it has the potential to fold into two different stem loop hairpins. And so how does this work? So this works because this leader region has the ability to form these stem loop structures. And so here we see what this leader region is able to do. So it kind of has these complementary regions. So whenever there's complementary regions that allow you to form 5' anti-parallel, sorry, I think I'm getting this wrong, but the idea is that you can form this anti-parallel binding, so you have complementary nucleotides and then they form in this anti-parallel way. And so in this way you can produce these stem loop structures. So there's the stem, where this binding occurs, and then there's the loop structure here. So you see multiple different stem loop structures that are occurring. And so what happens is that a ribosome is able to bind, and there's these key tryptophan codons. And so whether or not this ribosome can actually process and move through this mRNA is dependent upon the availability of tryptophan. So the ribosome is only able to move through if it's able to incorporate the tRNAs that hold the tryptophans. And so again, this is another way to incorporate the presence or the absence of tryptophan. So if tryptophan is around, then this ribosome is able to read through without a problem. If it stalls, then it's unable to go through, and then the ribosome stalls because it's waiting for the tRNAs to come by that has the tryptophan tRNAs, the charged tRNAs that have tryptophan on it. And so this stalling or proceeding basically creates an alternative stem loop structure. So in one case you have a stem loop structure where this 2 and this 3 region are bound together. And then in another case you have a situation where this region right here is bound to here. And so this movement through this separates the 2 and the 3 region, and it allows the formation of a new stem loop structure. So in one case, this stem loop structure is able to terminate transcription. So instead of the mRNA continuing to be able to read through, it simply acts as the stem loop, row independent mechanism of transcription termination. So the extension of transcription is stalled because of the action of an early transcription terminator. Whereas in the other process, if it stalls, then this stem loop structure is unable to form, and then the mRNA is able to continue along. So this is what is referred to as attenuation. It's the idea that the action of the ribosomes can modify the secondary structure of the mRNA. And this secondary structure of the mRNA influences whether or not transcription proceeds or not. So attenuation is not that common, but it does seem to be very common for the synthesis of a number of amino acids. So obviously it makes sense that this is a mechanism that kind of only makes sense with amino acids, because the amino acids presence can be translated into tRNAs being present or not. So it's been shown to also be important for threonine, histidine, leucine, and phenylalanine. But the general idea of mRNA secondary structure being important has led to kind of this idea of riboswitches. And so riboswitches have alternate forms of mRNA secondary structure. And so this alternative formation either creates an anti-terminator or a terminator structure. So the mRNA either terminates early before it has transcribed the genes, or it is able to kind of extend throughout the entire operon and create the genes. And so, for the tryptophan, this secondary structure, what controlled it was the ribosome. So the ability of the ribosome to move through or not to move through the leader sequence. But it's also possible to use small ligands. And so the small ligands are able to influence the secondary structure. And so these ligands actually can bind directly to the mRNA molecule itself. So part of this mRNA molecule contains a ligand binding region. And so if this ligand is not present, then we have this first secondary structure. If the ligand is present, then it has this secondary structure. So in these two cases, you either have transcription continue or transcription stop, depending on the presence or absence of a ligand. So these are called riboswitches in the sense that there's actual parts of the RNA sequence that can sense a particular metabolite. So there's two important domains within that riboswitch. So one is the aptamer. So the aptamer is the part of the mRNA that actually binds to the ligand. And then there's the expression platform, which is the part of the mRNA that is able to create this terminator structure to stop transcription. So that is the extent of what we're going to cover in this chapter. So we're covering some really key concepts. So these are just examples, and the purpose of this chapter is able to not just understand, the details of the lac operon, the details of the TrpR, but the goal of this is to give you some kind of general principles of how transcription can be regulated. And so there's all sorts of different ways that transcription is regulated, but in general, some of the main ideas are that you have DNA binding proteins. So these are proteins that bind to either the promoter or nearby promoter regions, and these turn on or off expression of a gene. And these proteins are often allosterically regulated. So they can be allosterically regulated by the binding of lactose. They can be regulated by the binding of secondary molecules. But there's also other ways to bind to regulate these as well. So phosphorylation is kind of a common way where a protein can have specific sites, specific amino acids phosphorylated, and that can cause a conformational change. So the allostery is also a really important principle of this chapter.